Hello, my name is Ebony Flowers. I am a postdoctoral researcher at the University of Southern California, and the title of my talk is The Impact of Cellular Stress on Cancer Cells. My postdoctoral research examines the various forms of cellular stress. We all know how stress can cause psychological changes, but how does stress impact the cells inside your body? Like you, your cells must go through constantly changing and stressful environments. For most cells, too much stress leads to cellular damage and ultimately cell death. In the case of breast cancer, however, cellular stress can be repurposed to enhance tumor growth and promote the spread of cancer to other organs. My studies explore how breast cancer cells utilize cellular stress for their benefit. In order to perform properly, your body's biological and chemical actions must be constantly monitored and controlled. When something goes wrong, these tightly regulated systems can become overloaded, causing cellular strain or injury. Depending on the source, duration, and severity of the stressor, cells must adapt accordingly. These outcomes range from protective mechanisms to promote survival to intentional death programs when cells are damaged beyond repair. One of the most common forms of cellular stress occurs when there are not enough nutrients available in the body. Most cells rely on glucose and amino acids to provide the requirements for many biological activities. But when these resources are low, cells often use reserves of fat or scavenge what they need from their surrounding environment. Other limiting factors that directly impact cells are low oxygen, known as hypoxia, and lack of space that can compress or stretch cells beyond their normal structure. Sometimes it is the routine activities of the cells that causes cellular stress. While your cells are working, they often generate byproducts known as reactive oxygen species, or ROS molecules, that are highly unstable. Under normal circumstances, these molecules participate in a beneficial capacity within the cell. However, under stress conditions, ROS molecules accumulate, and their instability can lead to cellular damage and ultimately cell death. Even well-accepted conventional treatments can be detrimental to cells. Chemotherapy and radiation, two of the most utilized treatments for cancer patients, can often cause unintentional damage to normal cells. These therapies are designed to attack tumor cells that quickly divide and grow. But your body also has other groups of cells with high turnover, including intestinal cells, skin and hair cells, and numerous cells of the immune system that can become collateral damage when trying to treat a chronic condition like breast cancer. Despite all the ways your cells can be compromised or injured, your body has developed several approaches to counteract these injuries. Unfortunately, cancer cells have found a way to hijack these mechanisms for their benefit. Having a better understanding of not only the inner workings of these cells, but how they interact with their environment is vital to finding better ways to detect and treat diseases like breast cancer. With comprehensive studies like this, we will better be able to exploit unique characteristics of cancer cells without harming any of the normal tissue. And to help accomplish this objective, my research investigates how breast cancer cells that leave the primary tumor give rise to new tumors in other organs. The cells that accomplish this are called circulating tumor cells, and I study how they are able to survive the taxing departure from the mammary site, travel through the bloodstream, and give rise to new tumors in organs with completely different microenvironments. This research will expand the knowledge we have in the complicated network of cancer biology, improve diagnostic and therapeutic techniques, and most importantly, help in the pursuit of improving the lives of cancer patients.